So, into the last chapter on lab techniques, this time involving DNA. Two people walking down the street surrounded by, there's a blob wearing a hat, other blobs. Remember when there was all that fuss about recombinant DNA? I'm not entirely sure how much sense that makes, but when viruses invade bacteria, one of the defenses that bacteria have is an ability to take the DNA that is being imported from the viruses. Remember, that's what the virus is going to use to make more viruses. And they break it up. They literally snip it to pieces so that the viruses can't actively make more viruses. They have a set of enzymes where the active sites attach to stretches of DNA that are not found in the bacteria, but are found in the viruses. So they're kind of always there ready to snip in particular stretches of DNA. And these are called restriction enzymes. In order to run electrophoresis on DNA, you're going to migrate it through usually a gel, and you're going to clip it into pieces that are different sizes. Because it's all DNA, the positivity, negativity of it is pretty much always the same. So all you're going to do to separate is migrate stuff by size. And because you're using restriction enzymes, there's certain predictable snippets that you're going to get when you clip it. So what we've got here, this is forensics. So you've got DNA from a crime scene, DNA from a suspect and another suspect. Well, you can get enough of a sample from a suspect to run straight up testing. From a crime scene, a lot of times you don't get enough and you need to amplify it. We'll talk about that a little bit later. And so you use PCR to make enough DNA that you can then migrate it through your gel. And then you look to match the bands to someone. Typically, you're not gonna, I think the total number of bands that you typically get is a little bit more than this, where you need to match something like 75% of the bands from your sample to your suspect. But as you can imagine, you're not matching the genome, you're not matching the sequences, you're matching bits and pieces. And so the odds of you absolutely positively getting a match that you can be absolutely positively sure doesn't happen that often. I mean, you don't normally, even when you have a crime scene matched against a suspect, you often don't line up every single band for various reasons. As the DNA degrades on the scene, it doesn't always, it's ANSI in these processes. It doesn't always do what you want it to do. So usually there's a threshold of, if we can match these many bands, we're going to consider it a match. But then they'll go into court and go, oh yeah, it's like 1 in 40 million chance that it was a mismatch. And number one, they're making those numbers up because nobody's actually run that data so far as I've ever seen. And it's reliable, but it's not as reliable as they sell it. I mean, the same thing's true for fingerprints. His fingerprints are not as reliable as they look like on TV shows. But a lot of times what happens is you run a fingerprint through a very large database, and based upon the bits and pieces of a fingerprint that the computer looks for, it may give you like 10 matches. And you look and like okay, these 3 people of the 10 matches are dead, these 3 people are on the other coast, these 2 are out of the country, this is from the Interpol database, and okay, here's someone who actually lives here. That's probably who it is. But the idea that you run it through the thing and you get like one person pop out of the computer, a lot of times that's not how it works. And when we have DNA databases, that may also be true. But this is how they do it. Give you a somewhat older technique, something called the Southern Blot. With the Southern Blot, you're trying to find particular gene sequences. So what you do is, and this only works in if you've got cells that are making a lot of the target protein. Because what you do is you take the active cells and you extract the messenger RNA that they're using to make that particular sequence. And then you run electrophoresis. You take your mRNA and put a radioactive marker on it, and you dose your gel with the radioactive tagged mRNA. And then, actually the old technique, and I don't know how much they still use it, is you would take your gel and make a sandwich of it with undeveloped film, photographic film. Do that overnight, and the radioactive tags would make a light band on the film and show you which band has the piece of DNA that you're looking for. And with successive clips, you can actually find a tiny stretch of DNA that is the gene that you're looking for. The protein is being made in this cell by a gene. It does that through mRNA to get there. And so you're gradually finding where it is. And then you can analyze the gene sequence itself. There are variations. Southern Blot was developed by a fellow named Southern, but as they put new variations out, people go, well, we're gonna call this Northern Blot, or this Western Blot, or this Eastern Blot. So there are variations that have nothing to do with actual directions. It was just that the first person was named Southern, so people have gotten kind of funny in developing new techniques. This has revolutionized DNA testing. Used to be, if you wanted to match DNA from a crime scene, you needed a big pool of blood or a body of someone that if you thought they were related to somebody, but it wasn't obvious that you might be able to, you needed a lot of sample. There was no way of taking a small amount of DNA and making lots and lots and lots of copies of it. Well, that's what PCR does. It's called an amplification technique. And it was developed by a fellow who made lots of money on it for him and his university. He was studying Thermus aquaticus, a bacterium that lives in hot springs. When you live in virtually boiling water, or adjacent to virtually boiling water, then you better have some pretty robust enzymes for doing stuff, including making DNA copies. And so he took what's called TAC, Thermus aquaticus, TAQ, TAC polymerase, the DNA producing enzyme, DNA polymerase. And he developed a technique where you could take a small amount of DNA and make lots of copies, not of the entire genome, but of, again, bits and pieces of the DNA. Some people have been able to do it with entire genomes, but it's very, very tricky to do it with entire genomes. So here's what happens. You take your small sample, you've got a little bit of DNA, and you put it in your machine, and you heat it up. I have not a good memory. As you know, I don't have a good memory for numbers, so I'm looking at my numbers here on the sheet. You heat it up to just south of boiling, 94 to 95 degrees Celsius for a few minutes, 2 to 10 minutes. And what happens is the DNA molecules separate, right? They can't stay, it's hydrogen bonds that keep your two strands connected. You shake those and now you get single strands of DNA. You lower the temperature down to 50 to 60 degrees Celsius. Now, okay, what does that mean? Well, 37 degrees is body temperature, so you're like, say 15 degrees above that, 15 thereabouts, which means you're about 30 degrees Fahrenheit above body temperature, so you're up in the 120, 130 degree range. It's pretty doggone, most hot tap water runs in that range of, in fact, a little cooler. So it's pretty hot. And so you lower the temperature, and what you've done is you've mixed in a bunch of primers, little pieces of single-strand DNA that will clip onto the DNA from your sample at a particular spot and clip on to your DNA at another spot. And what you're going to do is build a new strand between the primers. So you not only can amplify the DNA, but with the right kind of primers, you can amplify a particular part of the DNA. If you want lots and lots of copies of a particular gene or a particular part of the genome in order to analyze or use, you can do that by, and there's a whole companies devoted to making the primers. And that's where it gets a little tricky, is that the primers attach where you want them to attach, but they also, a certain level of them, a certain amount of them will attach someplace that isn't exactly a fit, but it'll fit there anyway. So there's limits to how many times you can run this and get good results, and different types of stretches will give you more or fewer mistakes. You can actually fit it to particular alleles though, particular variations. So you heat it back up, not to the first level, you heat it up to between 68 and 72 degrees Celsius. So you're between 50 and 60, now you're going up to between 68 and around 70. So you're going up, another 20, 30 degrees Fahrenheit, something like that. See, this is why knowing the ballparks is useful. And with the find the temperature, I needed a crossover point that I understood, which was body temperature. For a temperature increase, I just need to know that a Celsius degree is roughly twice as big as a Fahrenheit degree. It's technically 1.8, but for ballpoint, you can go to 2. Why do you warm it back up? Because that's the optimum temperature for Taq polymerase. It works in hot springs, so it works at pretty toasty levels. So we're talking 150 to 170 degrees Fahrenheit, somewhere around there. And it goes to work. It makes a new strand of DNA between the front and the reverse primers that you put in there. And so it makes copies of just that part of the DNA. So now you've got a copy of all the DNA molecules you put in there, but you need more than that. So you let it cook for 10 to 30 seconds. You're not making really huge DNA copies, so you don't need to leave it doing this for very long for it to make what you need to make it. If you leave it for too long, sometimes you get more stuff than you really wanted. And then you raise the temperature back up, get the strands to disconnect, bring the temperature back down, throw the primers on, raise the temperature to the optimum temperature for the polymerase, make copies, raise it up, get the copies apart, put it back down, put more primers on, raise it, make copies, and it cycles. Is that you throw all this stuff in enough to run a bunch of cycles. Remember, enzymes are reusable, but the primers are not reusable. So it goes, and basically this is done in a machine. As I went to Bethesda many years ago, well, not that many years ago, and learned in the labs, the National Institute of Health labs, how to do this. Ran the machines. The machines are even more complex now than they were then, but I got a basic grasp of how this happens. So we ran lots of copies and did some, and then to make sure we were getting what we wanted, we ran electrophoresis and looked at the bands. We knew what bands we were trying to get to find out whether we were actually doing it well. It turns out my daughter, who went with me to learn this also, I'm a bit of a slob, I'm somewhat sloppy in the lab, as I often don't do things as precisely as I probably should. I'm a parasite biologist, We cut things up and pull worms out. She got, until the very last day where we got our final results, she tended to do way better because she's much more precise in the lab than I am. But I got better results at the end. Because then I knew it really counted. So after a while, you kind of settle it, and then you cool it down to 4 degrees Celsius. Remember, that's about 40 degrees Fahrenheit. And that's where you let it settle, is that nothing's really happening except what you've got is stabilized at that temperature. Now you can also treat RNAs and basically use reverse transcriptase to make DNA out of it, and then do this to make lots of copies of the DNA that you can then analyze. That's what's done with the up-the-nose COVID test. What they call, it's a PCR test. But what COVID has is RNA as its genetic payload. And you can't make straight up copies of RNA. I think somebody's developing a way to do that. I seem to remember reading about that. But so far as right now, not widely used, unless they're doing it on the COVID test. I don't believe they are. I believe they're using reverse transcriptase to make DNA, and then they're amplifying the holy hell out of the DNA really fast and checking it for the viral genes. And that is a good place to pause, to stop before we do the next video here.